My name is Joachim Schiemann. I'm head of the Institute for Biosafety of Genetically Modified Plants, which belongs to the Julius Kühn Research Center in Germany. I'm professor at Lüneburg University. I'm a member of the GMO panel of the European Food Safety Authority, and I have been just recently 4 years president of the International Society for Biosafety Research. So we are 9 partners in our work package 1, representing 5 different countries: Germany, Switzerland, Bulgaria, UK and France. And our partners are coming from basic research, from applied research, from breeding companies and also seed producing companies. So our work package has 2 main objectives. One objective deals with biological mitigation techniques, and the other objective deals with drivers for pollen-mediated gene flow in maize. So I will start with the second part, which is a smaller part. We have been developing models to predict gene flow, also gene flow over fragmented landscapes, which might help the stakeholders to develop more flexibility. So nowadays, in different member states, we have fixed isolation distances between neighboring maize fields. To use these models, where a lot of factors are included, like field size, distance between the fields, wind velocity, main wind direction, flowering coincidence, and a lot of other factors, might help to make the system more flexible. Another part of that main objective is to look for admixture. So the question is, when we have a specific admixture in the seed material, do we find a similar admixture in the harvest? To answer that question, we performed ring experiments in different member states under different climatic conditions, field management conditions, and so on. And we were very happy to find that, let's say, when you have 1% admixture in your seed material, you find, under all the different conditions, about 1% in your harvest. So the main part of our research is to look for the stability and usefulness of different biological mitigation techniques. That means to reduce the pollen-mediated gene flow by some biological means. We are dealing with 3 mitigation techniques. One is to introduce the transgene not into the nucleus of the plant, but into the chloroplastic genome. This approach has the advantage that a lot of plants do not inherit the chloroplasts and the mitochondria paternally, so they are only inherited by the mother, and the pollen does not contain chloroplasts and mitochondria. And one of our partners has been testing the reliability of that method, and he found very good results that in the case of tobacco, which has been used as a model plant, we find very, very high stability of that mitigation technique. We also have been looking into the stability of 2 other biological mitigation tools. One is the cytoplasmic male sterility in maize. The other is the cleistogamy in oilseed rape. So in the case of cytoplasmic male sterility in maize, we have been performing quite extensive ring experiments, and we have been testing several lines belonging to 3 different types of male sterility. And we found that cytoplasmic male sterility is a quite useful method to restrict pollen-mediated gene flow. And I think as a very good outcome for the stakeholders, we can say that in all 3 types of cytoplasmic male sterility, we found some lines with a very high stability, also lines with medium stability and also some lines with lower stability. So it might be used by breeders as a mitigation technique depending on the different traits which should be introduced in maize. And you can use it as a tool, for example, to reduce the isolation distance or to produce specific products in maize, including, for example, industrial products or pharmaceutical products or others. The other topic we have been testing in several ring experiments is the cleistogamy in oilseed rape. Cleistogamy means that the flowers do not open, so that there is no pollen flow out of that plants, of that flowers, but also not a pollen flow inside these plants, inside these flowers. And we have tested two different oilseed rape lines, which are cleistogamic. And we found that one line is very stable, so the reduction of pollen flow is by 90%, 95%. And also this technology might be used by breeders to link the cleistogamy with specific traits which might be introduced into oilseed rape. So at the end, I'm quite happy about our results. There are several programs, research projects going on in different member states, and I think it was a very good outcome of the EU-funded projects that all these national activities have been combined, that we exchanged protocols, different testing methodologies. So we have been performing a lot of ring experiments, and so I think that there is really a very good added value on the basis of the EU-funded project.